Hello everyone, welcome you all in our YouTube channel Vaishnavi Nalaude. In today's video lecture, we will discuss about remaining part of our third chapter in plant tissue culture in Pharmacognosy and Phytochemistry 1 for B-PHARM. In that, we will discuss the aseptic condition in the basic requirements of plant tissue culture. ओके, हा पार्ट आपण काल बघितलेला आहे. इन aseptic conditions, in first part we will discussed about the aseptic conditions in basic requirements of plant tissue culture. Okay, the next one, that is washing and storage facilities. वॉशिंग अँड स्टोरेज फॅसिलिटीजमध्ये काय आहे, त्याच्यामध्ये फ्रेश वॉटर सप्लाय अँड डिस्पोजल ऑफ वेस्ट वॉटर फॅसिलिटी शुड बी अवेलेबल. Space for distillation unit for the supply of distilled and double distilled water and deionized water should be available. Working table, sink or wash basin for apparatus or equipments washing should be acid and alkali resistant. Sufficient space is required for releasing hot air oven, washing machine, pipette washers, etc. For storage of dried glassware, separate dust-proof cupboards or cabinets should be provided. These are the washing and storage facilities of equipments and apparatus. Okay. Then next one, that is media preparation room. It should be spacious to accommodate labware, culture vessels, equipments, chemicals, etc. The preparation room should also be well equipped with refrigerator, freezer, etc. for storage of media and stock solutions. मीडिया प्रिपरेशन रूम ही काय असली पाहिजे, क्लीन असली पाहिजे, वेल इक्विप्ड असली पाहिजे आणि त्याच्यामध्ये आपण जो मीडिया आणि स्टॉक जे काही सोल्युशन्स करणार आहोत तेच कसं असलं पाहिजे, फ्री फ्रॉम मायक्रो ऑर्गॅनिझम्स. ओके, दीज आर basic requirements of plant tissue culture. Then next one, that is sterilization room. This is basic. In the tissue culture lab, it is desirable to have separate sterilization room for sterilization of culture media, glassware, metallic equipments like scissors, scalp, etc. Generally, sterilization is done in autoclave or hot air oven. Sterilization room, in the tissue culture lab, it is desirable to have separate sterilization room for sterilization of culture media, glassware, metallic equipments like scissors, scalp, etc. Generally, sterilization is done in autoclave or hot air oven. Then nutrient medium. In that, media is composed of inorganic nutrient, organic nutrients and other media substances. In that, inorganic nutrients, which includes micronutrients like nitrogen, phosphorus, potassium, calcium, etc. and micronutrients like boron, copper, iron and manganese and zinc. These are inorganic nutrients. Okay, then organic nutrients, in that it includes vitamins like vitamin B1, B6, B3, B5, etc. and amino acids like L-arginine, L-asparagine, L-cysteine, hydrochloride, hydrochloric acid, L-glutamine, etc. Carbon source like glucose or maltose, growth hormones or regulators like auxin, cytokinins and gibberellins, ethylene, abscisic acid. These are organic nutrients. Okay. Then other media substance, in that like protein hydrolyzed acetase, yeast extract, fruit like banana extract, coconut milk, solidifying agent like agar, alginate, gelatin, etc. Iron source, example in iron source, that is EDTA. Then antibiotics. These are other media substances. In pH, in that of the medium should be in a range of 5.6 to 6.0 before autoclaving the culture medium. Nutrient medium, in that media is composed of inorganic nutrient and organic nutrient, other media substances. pH, these are nutrient media. ओके, कोणकोणते न्यूट्रिएंट्स आपल्याला यूज करायचे आहेत. देन transfer room, in that it is provided with the laminar flow hood where most of the work of culture initiation and subsequent subculturing is performed. Culture replantation, transfer or reinitiation in a clean media, harvesting of ripe cultures is also performed in this area. These are transfer room. It is provided with the laminar flow hood where the most of the work of culture initiation and subsequent subculturing is performed. Culture, it is replantation, transfer or reinitiation in a clean media, harvesting of ripe cultures is also performed in this area. Then culture room or incubators. In that cultures are incubated on shelves or in incubators under specific condition of temperature, humidity, air circulation and light. Incubation chamber or area should have both light and temperature control devices managed for 24 hours period. Generally, high output, cool, white fluorescent light is preferred for a photo period duration, that is specific period for total darkness as well as for higher intensity light with a temperature range of 25 plus or minus 2 degree Celsius, range 18 to 25 degree Celsius. The rooms are required to be maintained at a humidity up to relative humidity up to 70 to 75% in the range of 20 to 90% controllable to plus or minus and uniform forced air circulation. These are culture room or incubatorचं टेम्परेचर कसं असलं पाहिजे. ओके, then proper and optimum aeration, in that adequate aeration is required for cell to grow. Tissues which are cultured on semi-solid media do not require any special method for aeration. But tissues which are grown in suspension cultures require special devices for aeration. Aeration for submerged cultures can be provided by following methods, in that placing the culture vessels with the liquid media on an automatic shaker. The two ends of the filter paper are dipped in a medium and the middle horizontal portion on which the tissue is placed remains above the level of the medium. This method is called as filter paper bridge method. Passing sterilized air through the medium and by stirring the medium. The culture vessels are closed with non-absorbent cotton covered in cheesecloth. This process allows proper aeration but prevents the entry of microorganisms. These are the proper and optimum aeration conditions for cell to grow. Okay. Then well-equipped observation or recording area. In that at regular intervals, growth and maintenance of the tissue culture in the incubator should be recorded. All observations should be done in aseptic environment. At regular intervals, growth and maintenance of the tissue culture in the incubator should be recorded. All observations should be done in aseptic environment. For microscopic examination, separate dust-free space should be marked for microscopic work. These are well-equipped observation or recording area for growth. Okay, then these all basic requirements of plant tissue culture. Okay, The next point in that basic methodology or techniques of plant tissue culture. In that the general technique used in the isolation and growth of culture is described as follows. In that first, preparation of suitable nutrient medium as per the selection of plant medium is autoclaved. Then second one, that is selection of explant, in that any excised part of health plant to be used. Example, that is bud, leaf, root or seed. Then sterilization of explant, in that by sodium hypochlorite, mercury chloride, etc., and washed aseptically for 60 to 6 to 10 minutes with sterilized water. Then inoculation, that is transfer. The sterile explant is inoculated on solidified nutrient medium under aseptic condition. Inoculation, in that transfer. The sterile explant is inoculated on solidified nutrient medium under aseptic conditions. Then next one आहे incubation. In that incubation मध्ये काय आहे तर कल्चर्स आर incubated at of 25 plus or minus 2 degree Celsius and at a relative humidity up to 50 to 70% from 16 hours of photo period. Then next one, regeneration, in that plantlets regenerated after transferring a portion of callus into another medium and incubation of roots and shoots are or directly from explants. Then hardening, in that is the gradual exposure of plantlets for the acid sorry hardening, in that is the gradual exposure of plantlets for acclimation to environment conditions. Then plantlet transfer, in that plantlet are transferred to greenhouse or field conditions. These are the basic methodology or techniques or steps in the plant tissue culture. First, preparation of suitable nutrient medium, selection of explant, then sterilization of explant, then inoculation, then incubation, regeneration, hardening and plantlet transfer. These are the procedure, basic methods or steps in the plant tissue culture. You can see in that first, that is field, plant in field, then washing, then surface sterilization with sodium hypochlorite in the trimming in sterile medium, then several washes, distilled water, then transfer to that test tube, in that inoculation or final trimming and establishment of cultures in that stem, that is explant, and medium, that is MS. Then callus preparation form, in that incubation at 25 plus or minus 2 degrees Celsius to 50 to 70% relative humidity for 16 hours. Then formation of सर्फ कल्चरिंग. ओके, 3 टेस्ट ट्यूबमध्ये ट्रान्सफर करायचं. जे कॅलस असणार आहे, बॉडी मास सेल मास असणार आहे, ओके, ते तिन्ही याच्यामध्ये ट्रान्सफर करायचं. देन सब कल्चरिंग, इन दॅट बर्ड्स आर फॉर्म, देन मायक्रोशूट्स आर फॉर्म, देन प्लांटलेट अँड रूट इज फॉर्म अँड देन ट्रान्सफर टू ग्रीन हाऊस, देन अटॅच पॉलिथिलीन बॅग, देन preparation of new plant. Okay, these are the procedures included in the plant tissue culture. Okay, then these are the axillary branching, in that parent plant to rooting. Okay, then parent plant to formation of callus, then shoot differentiation, then formation of rooting. There in that third method, in that adventitious budding and that form rooting. Then in that parent plant form, then from that callus formation, then formation of cell suspension, then somatic embryogenesis, then germinating embryo, and that form transplantation. Okay, these are the various methods included in the plant tissue culture, in the selection of plant. Okay, then preparation of an explant, then inoculation, then after incubation, then various stages of plant growth, then plant ready to be transferred into greenhouse or hardening stage, then regeneration of a plant from an explant. These are the techniques or steps in the plant tissue culture. Okay, then these are laminar airflow bench. ओके, इथे तुम्हाला दिसेल ह्या चित्रामध्ये की इथे असेप्टिक कंडिशन्स मध्ये हे काय आहे, दोघ जण काम करत आहेत. ओके, सो आपल्या मायक्रोच्या लॅबमध्ये सुद्धा असाच असेप्टिक लॅमिनार एअरफ्लो बेंच आहे. ओके, these आर टिश्यू कल्चर रॅक. ओके, मोठ्या प्रमाणात जिथे कल्टिवेशन केलं जातं त्या तिथे काय केलं जातं, टिश्यू कल्चर टेक्निक्स यूज केल्या जातात इन दॅट असेप्टिक कंडिशन्स फॉलोड बाय दॅट पर्सन. ओके, देन नेक्स्ट वन दॅट इज टाइप्स ऑफ कल्चर. हे आपण नेक्स्ट लेक्चरमध्ये बघणार आहोत. ओके, सो टुडे वी विल स्टॉप हियर. थँक यू सो मच फॉर फुल्ली वॉचिंग दिस व्हिडिओ. Happy learning, stay safe. If you have not subscribed to my channel, then subscribe first and press the bell icon to get latest updates of upcoming videos. Thank you.